Hello everyone, my name is Zheng Pang. Today we are talking about the experiment is compare the fermentation efficient between corn kernel extract and stem extract. Before we starting, I am very thanks for my lab partner Kaiki Kobayashi for helping to do this experiment. So we are starting off the overview, and for the background, nowadays the petroleum will be used in many different places in the world, and so we will also get increased interesting into the sustainable production, which is the biofuels. And for the biofuels, we were looking at how the products produce the ethanol in fermentation. And so in the fermentation, it will require glucose. For the glucose processing, it will showing by this picture. And the glucose will be start at glycolysis to 2-pyruvate into the cytosol. And after that, it will have two different steps. The first one is the red square one. It will including the oxygen and into the mitochondria to do the stellar respiration. And the second one will be the green square. It will be without oxygen and produce the 2-ethanol at the end. So the first one is the presence of oxygen, and the second one is absence of oxygen. And as you can see into the 2-ethanol producing, it will without oxygen, so the oxygen is not required in the fermentation. So in the experiment design, we will have 4 treatments in our experiment, and both treatment have 7 ml of total. And the first and fourth one will be the control group, and we will focusing on the second and third one, which is the experiment group. So starting for the experiment, we will get 5 ml substrate in each tube, and we will place the tube in the 30 degrees temperature of water bath for 10 minutes because the 30-degree temperature is the higher efficient for yeast. And here's the picture about the 4 tube. And we will add yeast when the 4 tube achieve the 30-degree temperature. And we will vigorously swallow the flask contain the yeast because we will make sure the yeast will be resuspend. And we will add 2 ml yeast in the tube. And also we will vortex the tube to mix the liquid. And we will get the mixed liquid to fill the Durham tube. And we will invert the Durham tube and place it to the original tube. So this is what we do. And when after the inverted Durham tube, it will have the maximum liquid into the Durham tube, and this is the both volume. And after that, we will return the tube to water bath again and record and start time and volume in Durham tube. And we will also repeat this step to other 3 tube again. And we will also record the volume in Durham tube after 5 minutes each until the 45 minutes. So here is the picture of you will might be see after the start time. So the carbon dioxide volume produced will be calculated by start time volume minus recent time volume, as the top space will be the carbon dioxide producing. And here is the result graph, which is the carbon dioxide volume produced in 5 minutes each. And you can see that the kernel extract in water have the fastest in fermentation, which is start at 10 minutes. And then we will also see that the stem extract in water plus yeast have a lower rate, and we will almost achieve the 0. So it will also can seem that the stem extract will have not do the fermentation. And also the corn syrup in water plus yeast have the higher amount of carbon dioxide produced in this graph. So because the stem extract and the buffer one have a lower slope, and is almost zero. So we will only calculate the first and second one, which is the corn syrup and the kernel extract. And for the second one, kernel extract, you can see it will have the 0.42 ml for each minute. So it is higher than the stem one. So we can get the result is kernel extract have higher efficient to fermentation than the stem. And also according to the Saylor's journal, it will have 66.8% of starch of dry material into the intact kernel. So it can seem that the kernel have the including more starch, and the more starch will be produce more glucose. And for the conclusion, the main part of corn fermentation, we can conclude that is in the kernel because it have the high rate of the fermentation. And the second one is the visible understand the fermentation processing for me to help me to understand how is the fermentation processing. And the third one is it will improve in the future, the scientists will be improve some products to get more ethanol, like build some new type of corns can produce more ethanol in the future. And that's all. Thank you for your watching. And before we ending, I will thanks again for my lab partner, Kaiki Kobayashi. He is very friendly and helpful for me to do this experiment. And here is the reference. And that's all. Thank you.